Okay, sometimes we're dealing with things that we need to get in and out of the cell that are too big to kind of just go through the membrane or to even go through a transporter. So everything we've dealt with so far has been pretty small. So like ion, sodium, potassium, even water. But a lot of times cells bring in things that are much larger than the small molecules that we've talked about so far. So the one process that we want to, oops, scribble in here for no reason. The first process we want to learn about is how cells get large amounts of material into the cell, and that's endocytosis. Into the cell. And what they basically do is they ingest this is like an ingestion of external materials by folding the membrane around them to form a pouch. We'll show you pictures and animations of this in a minute. They form a pouch. And then this pouch will then pinch off and becomes a vesicle. We learned about in chapter 4 what vesicles were. Vesicles were just a membrane-bound circle or sac that brings things around the cell. Remember, vesicles came off of the Golgi apparatus. Vesicles came off of the endoplasmic reticulum. So vesicle is just something that holds something. So a lysosome is an example of a vesicle. Okay, there's two types of endocytosis, and I'm not sure why this isn't all showing up here. Okay, the first type of endocytosis is called pinocytosis. Pinocytosis is a process by which large amounts of fluids or things that are dissolved in fluids, so things that are dissolved, such as salts, sugars, even proteins that are dissolved, they're solutes in fluids. That process, which brings large amounts of fluids into the cell, is called pinocytosis or pinocytosis. And that process, you can kind of think of it, I like to think of it, if I use an analogy of the cell is kind of like it's drinking, because it's not going to be bringing solid materials into the cell. Everything is liquid or dissolved in liquid. And so that's the first part of endocytosis. And remember, we're talking about active transport here. So this process is going to require energy in order for this to happen. And pinocytosis is the process by which large amounts of fluids or solutes are brought into the cell. And that can be contrasted with phago or phagocytosis. Different people pronounce it different ways, just like pino or pinocytosis, in which this is a process by which large particles of things, solids, or even cells. So, for example, your white blood cells undergo a process called phagocytosis in order to fight infections, to fight bacterial infections, to fight viruses, large particles, cells. So that process I like to think of as eating almost. If we think of pinocytosis as drinking, phagocytosis is the process by which large particles are brought into the cell. And here's an animation.
During endocytosis, the cell membrane surrounds material and encloses it in a vesicle. The cell comes into contact with a particle. The cell membrane forms a pouch, and the material fills the pouch. As the vesicle fills, it moves laterally across the cell membrane. Once the vesicle is surrounded, a vesicle pinches off into the cytosol. At this point, the vesicle may fuse with a lysosome, as in the case of phagocytosis. If the vesicle contains a fluid, the endocytosis is known as pinocytosis.
Okay, so endocytosis in the form of either pinocytosis, which is bringing liquids or solutes into the cell, or phagocytosis, which is bringing solids into the cell. Endocytosis, as its name implies, brings things into the cell. Endo, in. Now, sometimes you have to get rid of large amounts of materials, and you want to be able to do that efficiently. And that is the process called exocytosis. And that just basically means to exit the cell. And instead of bringing something in, what's going to happen is the vesicle is going to go to the membrane. It's going to fuse with the membrane and then releases the materials to the outside environment. So that the materials are then released to the outside. So exocytosis, vesicles fuse with the membrane and then basically dump their contents out of the cell. And here's another animation showing exocytosis.
During exocytosis, a vesicle is moved through the cytosol to the plasma membrane. When the vesicle and plasma membrane come into contact, the two membranes fuse to become continuous. The contents of the vesicle spill to the outside of the cell. The materials are now outside of the cell. The outer plasma membrane regains its original shape.
Okay, I think you have this in your packet, but make sure if you see these pictures, this showing something going into the cell, and we're talking about large particles here, so this would be phagocytosis. We don't see just a liquid. If this said it was dissolved or if it was a liquid, then it would be pinocytosis. And then the opposite of that is when these are moving to the membrane and exiting the cell. You need to be able to recognize that as exocytosis. And we really just have one form of exocytosis. There's not like in endocytosis we have pinocytosis, which is for liquids, and phagocytosis, which is for solids. We just call it exocytosis for things that are Okay, here's one of our animations, and this will compare and contrast endocytosis with exocytosis.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first...
I really want you to know the two forms, endocytosis of phagocytosis, bringing solid materials in, and pinocytosis is liquid materials. So those are the two that you're responsible for, but watch this because this is how your body transports cholesterol and things like that, so it's interesting. But as far as what you need to remember for endocytosis, remember the first two, phagocytosis and pinocytosis.
Bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.